Video Tutorial 1, Overview and our first project. In this video we will describe the main objective of tappAS and how to create our first project with the correct input data. So, what is tappAS? tappAS is an application for the analysis of RNA sequence data down to the isoform level. It provides a set of data analyses, visualization and filtering to study the alternative splicing. What makes tappAS different from other tools is that it incorporates the functional perspective to the study of alternative splicing. tappAS does this by offering a set of novel analysis tools that reveal functional differences associated with differential isoform usage. Using this analysis tools, tappAS reveals changes in the inclusion of functional motifs and domains arise from alternative polyadenylation and alternative splicing. You can see tappAS functionalities in additional video tutorials. In this first tutorial, we will just help you get started with the application. Now, we will demonstrate how to create our first project in tappAS and which files we need. Basically, there are 3 input data files required to create a project and 2 options which will be covered in more detail below. These files are, an experimental design, a transcript-level raw counts expression matrix and an annotation feature file for our species. Experiment design. This file represents the structure of our experiment. tappAS support 3 different types of experimental design that include, case control, time course single series and time course multiple series. The structure of the different cases is showed on screen. You should know that the first experimental group is considered the control group. And, for both times course series, we need at least 2 replicates for a time slot. If you still have doubts, you can find more information on our website tappAS.org. The next file is the expression matrix that must contain raw expression counts for transcript-level expression and for 1 or more experimental groups. An advantage is that you can load an expression matrix with all the groups, but in the design file just select one of them, to run a single time course series, for example. The last file is the annotation feature. The annotation feature file contains very detailed annotation data for each transcript, both at the coding and non-coding parts of the sequence. Following a structure as shown on the screen. tappAS has the annotation files for 5 species, Arabidopsis thaliana, Drosophila melanogaster, Homo sapiens, Mus musculus and Z. mays. Also, tappAS has a Mus musculus demo with its own design and expression matrix, so, you can try tappAS if you don't have your own data. tappAS allows you to load your own annotation file, but we recommend you to use one of ours. Again, you can find more information on our website tappAS.org. Finally, there are 2 more options to create your project. You can use a low counts and coefficient of variation filter for removing transcripts with low expression levels and a transcripts filter for removing unwanted transcripts from a specific list file. Now, we will create a new project using tappAS. First, we need to open tappAS and click in New Project or go to Projects, and click on New Project. A window will open where you can upload all the files mentioned before. We need to assign a name to this project, since we will use a human sample, and name it as Tutorial Homo sapiens. The next option we need to select is the annotation feature file. In this case, we select Homo sapiens Ensemble 86. As we said, tappAS has 5 different species with their annotations and special option where you can upload your own annotation feature, but we select Homo sapiens. Next, we need to load our experimental design and our expression matrix. In this case, for this tutorial we are not working with real data, we have generated these files to show you how to create a project. As you can see, the files have the same structure we showed you, and you can see that we are working with a single time series because we use a unique group divided into different time points in our design. It is important to know that the expression matrix must have the same names as the design file. So, we need to specify that we are going to work with a single time series and we need to select the property option. Finally, we have 2 more options. First, a low count variant filter, which we will apply to the default value. In addition, a transcript filter is available if desired. Now, we can check if all is correct and create the project. The first time we use a new annotation feature file, we must download the annotations from our website. This is an automatic process, but depending on your internet connection, it will take a while. After that, tappAS is preparing the files to work properly. Depending on your computer, this can also take a while. When the process finishes, you are ready to use tappAS and all of its features. Before the video finishes, we will do a fast overview of tappAS to identify its structure and where the options and information are. At the center, in the upper panel data, you always see your data or the main information showed by the analysis. As you can see, tappAS shows you the information structure and link between transcripts, proteins and genes. At the top, you can see all different analyses you can run and see their results. These analyses are functional diversity analysis, differential expression and splicing analyses, feature-level differential splicing and differential polyadenylation analyses, and a set of enrichment analyses of isoform level. On the left side, you have a set of options to show more information about the data in the center of the screen. It can change depending the information that you are seeing, so better check it regularly. At the bottom, you have the visualization display. When you have created a new project, the first view you have is a general summary. You can see the quantity of transcripts, proteins and genes you have loaded. The number of transcripts and proteins per gene. The next chart you can see is a pie chart that shows the proportion of transcripts' structural categories. And the following next two graphs give information about the quantity of annotation sources per transcripts and genes. And a PCA plot about our groups based on our expressions levels. You can see how the program separates all the groups using two components. Finally, tappAS can visualize your gene annotation data, and we will show how doing it. For instance, we will select the gene KIF1B and we will show the aligned structure of the transcripts of a gene where you can see the functional annotation of transcripts. The next view is the protein view, and you can see the functional annotation of different features as domains, or motif. Another visualization is the genomic view where you can see genomic exon structure, or, as another example, the expression charts view where is showed the expression of our gene, our transcripts and the CD's expression of each one of them. In the next tutorials we will see all these sections deeper. Thank you for watching the video.